Immunoprecipitation, IP, from Creative Diagnostics. Immunoprecipitation is one of the most widely used antibody-based techniques. By targeting a known protein with a specific antibody, it may be possible to pull the entire protein complex out of solution. It is used to purify and enrich the protein of interest from a complex mixture. IP is an important step in many proteomics studies. There are mainly 4 types of immunoprecipitation. Individual protein immunoprecipitation. Individual protein immunoprecipitation is a procedure by using an antibody that is specific for a known protein to isolate that particular protein out of a solution containing many different proteins. Protein complex immunoprecipitation, co-IP. Co-immunoprecipitation is an extension of IP that is immunoprecipitation of intact protein complexes. Chromatin immunoprecipitation, CHIP. Chromatin immunoprecipitation, CHIP, is used to immunoprecipitate the protein DNA complex out of cellular lysates. Ribonucleoprotein immunoprecipitation, RIP. RNP immunoprecipitation, RIP, is a technique to immunoprecipitate the protein RNA complex using an antibody targeting the ribonucleoproteins of interest. The general protocol of IP can be outlined as following steps. Sample preparation. Pre-clear the lysate. Immunoprecipitation. Elution. First, sample preparation. For cell lysates preparation, harvest cells and wash with PBS. Resuspend the cell pellet in ice-cold cell lysis. Sonicate cells in ice bath. Centrifuge and collect the supernatant. For protein A-G agarose beads preparation, wash the beads twice with PBS. Dilute beads 1:1 with PBS. It is recommended to use wide orifice pipette tips or tips with the end cut off when manipulating agarose beads to avoid disruption of the beads. Pre-clearing the lysate is recommended to reduce the non-specific binding. Add pre-washed protein A-G agarose bead to the cell lysate. Incubate at 4 degrees Celsius for 10 minutes with gently agitation. And centrifuge at 3000 times G at 4 degrees Celsius for 2 minutes and collect the supernatant. Immunoprecipitation. Add relevant antibody to the pre-cleared cell lysate. And gently rock the mixture at 4 degrees Celsius for 2 to 4 hours or overnight. Add pre-washed protein A or G cephorose bead slurry to capture the immunocomplex. Gently rock the mixture at 4 degrees Celsius for 1 to 4 hours. Collect the beads by low-speed centrifugation at 2000 times G, 30s at 4 degrees Celsius. Wash the beads with cell lysis buffer or PBS and collect the beads by low-speed centrifugation at 4 degrees Celsius. At last, protein can be eluted from the beads by denature buffer or non-denature buffer, such as SDS buffer and glycine buffer. Besides, increasing the salt concentration in elution buffer will help to elute protein. And the eluent can be stored on ice for same-day analysis or frozen at -80 degrees Celsius for future analysis. The protein can be analyzed using the following methods: SDS page, Western blotting, gel band excision and sequencing, mass spectrometry, scintillation counter or X-ray film for radioactive samples, etc. Immunoprecipitation can be used in following areas: isolate, detect proteins of interest, enrich the low-abundant proteins, study protein-protein interaction in protein complexes, identify unknown proteins in protein complexes. Thanks for your view.